Welcome squad to another video. This is the 9700 October-November 2017 paper 1-2. If you are new to this channel, you know what to do. Like, comment, subscribe, and now let's watch and learn. Question 1. Which equation for calculating the actual size of a specimen A or image size I or magnification M is correct? First of all, you should know that the formula to calculate magnification is image size upon actual size. That is, M is equal to I over A. Always remember this formula. Magnification is nothing but the image size upon the actual size. Therefore, if you go to the answers A, B, C and D, the best answer over here and the only answer possible is answer D, because answer D itself, they have given straight away that magnification is image size upon actual size. Therefore, if you remember that formula, you can apply it in these kind of questions. Hence, once again, the answer is D. Question 2. One of the smallest viruses is the polio virus, which has a diameter of approximately 30 nanometers. In 2003, the Mimi virus was discovered, which has a diameter of approximately 680 nanometers. In 2013, the Pandora virus was discovered, which has a diameter of approximately 1000 nanometers, which row correctly matches a cell structure with a virus that has a similar approximate size. Once you see a question asking which cell structure has a similar size to a virus, first think about the size of a ribosome. The size of a ribosome is 25 nanometers. And which virus matches exactly with the size of a ribosome? That has to be the polio virus. And the only answer that has ribosome under polio virus is answer D. Nonetheless, let's look at answer D. Polio virus. Yes, ribosome does match with the size of a polio virus because polio virus has a diameter of approximately 30 nanometers, whereas a ribosome has around 25. So it is somewhat similar. And if you look at column Pandora virus, they have mentioned mitochondrion. And yes, mitochondria do have a size of around 0.5 to 1 micrometer, which is approximately around 1000 nanometers because 1 micrometer is equal to 1000 nanometers. Therefore, yes, mitochondria does have a similar approximate size to the Pandora virus. And even if you don't know what the other column size is, for example lysosome, since the other two are correct and it is the only possible answer, answering this question should be easy. In these kind of questions, if you don't know the size of an organelle, check out the sizes of the ones which you already know. So you have to remember the size of a ribosome, size of a mitochondria. These are easy to remember. Size of a ribosome around 25 nanometers, mitochondria around 0.5 to 1 micrometer, and nucleus is way too large. It is even beyond 1000 nanometers, therefore it is not possible as an answer over here. Therefore answer A is obviously incorrect. Nucleolus also cannot be the size of around 30 nanometers because nucleolus is larger than the ribosome. Ribosome has a size of 25 nanometers and matches with polio virus, whereas nucleolus is greater than the size of a ribosome, therefore it cannot match with the polio virus. Hence answers A, B, C are incorrect, leaving us with the only possible answer being D. Question 3. Which cell components are present in all prokaryotic cells? All prokaryotic cells obviously does not have endoplasmic reticulum. Therefore, the answer has to be either A or B. And not all prokaryotic cells have flagellum. Some do have, but it is not present in all. Since the word all is in bold, we have to meet this requirement. Flagellum is not present in all, it is present in some. Therefore, the best possible answer over here is answer B, not answer A, because answer A mentions that flagellum is present in all, which cannot be true. Hence the answer is B. Cell surface membrane, yes, is present in all. Cell wall, yes, all prokaryotic cells do have a cell wall. All prokaryotic cells do not have endoplasmic reticulum, and not all have flagellum, only some have. Therefore the answer is B. Question 4. The electron micrograph shows part of an animal cell. What will be synthesized in large quantities in this cell? Now, as you can see over here, the electron micrograph obviously shows the mitochondria and the nucleus. The mitochondria produces ATP, the nucleus makes nucleic acids, being DNA or RNA. Hence, inside this cell, only 1 and 3 are present in large quantities. We have to only look at this electron micrograph given over here. We cannot assume what are the organelles present in an animal cell. Just look at this given micrograph. You can see that there is a nucleus and there is lots of mitochondria. Mitochondria definitely produces ATP, as you all know. Therefore, 1 will be synthesized in large quantities in this cell. And the nucleus does make nucleic acids, which are either DNA or RNA. DNA or RNA are the nucleic acids. Therefore, 3, which is RNA, must be synthesized in large quantities in this cell. We do not know if glucose is synthesized in large quantities. Therefore, we cannot be certain over here because there is no organelle given over here that makes us think that glucose is synthesized in large quantities. Therefore, the answer is answer B, 1 and 3 only. Question 5. Mitochondria are thought to have evolved from prokaryotic cells that were ingested by an ancestral cell. Which feature have the prokaryotes lost during their evolution into mitochondria? As you obviously know, mitochondria do not have a cell wall, while all prokaryotic cells do. Hence, the obvious feature they have lost over here is a cell wall. Therefore, the feature the prokaryotics have lost during their evolution into mitochondria is cell wall. Answer A. Question 6. Which statement is correct? A. A virus is composed of a protein coat which may surround RNA or DNA. Well, yes, a virus is made up of a protein coat surrounding a genetic material which can be either DNA or RNA. Therefore, this statement does seem to be correct. Let's keep this statement aside for a while and consider the rest of the statements. Statement B. Eukaryotic plant cell walls contain peptidoglycans in addition to cellulose. Always remember that eukaryotic cells do not have peptidoglycan cell walls, they only have cellulose cell walls. Therefore, statement B is incorrect. Statement C. Plasmodesmata and centrioles are found in all plant cells. Centrioles are not found in all plant cells. You have to keep this point in your mind. Centrioles are not found in all plant cells. Therefore, statement C is incorrect. Statement D. Prokaryotic cells contain ATS ribosomes which they use to manufacture proteins. Prokaryotic cells, they only contain 7 TS ribosomes, not ATS. Therefore, statement D is also incorrect. Hence, we have no choice. The answer must be A. Virus is indeed made up of a protein coat surrounding a genetic material which is either DNA or RNA. Therefore, yes, statement A is correct. Question 7. A student carried out 4 tests for biological molecules on a sample of milk. The tests and their results were as follows. Heating to 80 degrees Celsius with Benedict solution gave a brick red color. Adding biuret solution gave a purple color. Adding iodine solution gave an orange color. Boiling with acid followed by neutralization, then heating to 80 degrees Celsius with Benedict solution gave a brick red color. Which biological molecules must be present in the milk? Well, the first test, heating to 80 degrees Celsius with Benedict solution, is a test for reducing sugar, and if it gives a brick red color, that is a positive test result indicating that reducing sugar must be present. Therefore, the answer should have reducing sugars. Let's look at the next test. Adding biuret solution gave a purple color. Biuret solution is for protein tests, and if it gives a purple color, that also indicates a positive test result. Positive result for protein test means that proteins are also present in the milk. Adding iodine solution gave an orange color. The iodine solution is used as the test for starch, and if it gives an orange color, that indicates it is a negative test result because the positive test result for iodine solution is blue-black. Remember that. So if it gave an orange color, that means it is a negative result for the starch test, indicating that there is no starch present. And the final test, boiling with acid followed by neutralization, then using Benedict solution. Well, boiling with acid followed by neutralization and heating is done for non-reducing sugars. And although a positive result is there for non-reducing sugar, we cannot be certain about it. because the non-reducing sugar test can give a positive result for the reducing sugar which was already present in test 1. Therefore, we have to consider that non-reducing sugars is not present in the milk, because non-reducing sugar test is only done if reducing sugar is not present, as a non-reducing sugar test shows the presence of non-reducing sugars. Therefore, if the test for reducing sugars gave a negative result, that is the first test, heating to 80 degrees Celsius with bendic solution gave a brick red color. If that didn't give a brick red color, that gives a color such as blue, that means reducing sugar is not present. And if that was the case, then we can be certain that non-reducing sugars must be present. However, over here, reducing sugar was already present in the milk. Therefore, the non-reducing sugar test can be influenced by the reducing sugar already present. Hence, the answer is answer C, 2 and 3 only. Question 8. Which features adapt a cellulose molecule for its function? 1. Long chains of glucose molecule coil into a helix. 2. Many hydrogen bonds form between adjacent chains. 3. It is insoluble in water. What are the features of cellulose? Cellulose has a high tensile strength due to the presence of many hydrogen bonds. Therefore, yes, many hydrogen bonds do form between adjacent chains. And cellulose forms cell walls in plants. You do know that, right? Plant cell walls are made up of cellulose, and therefore it must provide support to the plant. How does it provide support to the plant? By being insoluble in water. If it was soluble, it wouldn't be able to support the plant and keep the plant upright. In order to support the plant, it must be insoluble in water, as water pushes against the cell walls. Therefore, the features that adapt cellulose for its function is many hydrogen bonds form between adjacent chains, and it is insoluble in water. Therefore, the answer is C, 2 and 3 only. Why is answer 1 not included? Long chains of glucose molecules coil into a helix. Well, long chains of glucose molecules coiling into a helix can be for any molecule, and it doesn't have a specific purpose, or it is not related for its function. Therefore, it doesn't adapt a cellulose molecule to its function. Hence, we don't include answer 1. Only answer 2 and 3 are the realistic answers over here, because hydrogen bonds are necessary for the high tensile strength. Insoluble in water is necessary for forming cell walls in plants and providing support. Hence, the answer is C, 2 and 3 only. Question 9. Two disaccharides are maltose and sucrose. Maltose is formed from two molecules of glucose, while sucrose is formed from fructose and glucose. Which row shows a molecular formulae of the two disaccharides? You do know that glucose is C6H12O6, right? Therefore, you might think it should be either C or D. However, do keep this point in mind. When two glucose molecules combine due to condensation reactions, a water molecule is released. Hence, it becomes C12H22O11. So, if two glucose molecules combine, that is C6H12O6, combines with another C6H12O6, it forms C12H24O12. And if you minus a water molecule from that is, you take away two hydrogens and one oxygen, that leaves you with C12H22O11. Therefore, the answer must be between A and B. And remember, no matter the disaccharide, the molecular formula is the same for both. It is just the arrangement that differs. Hence, both of them, maltose and sucrose, should have the same numbers. And that is only there in answer A, C12H22O11, and for sucrose, C12H22O11. Hence, the answer over here is A. Answer B is incorrect because under sucrose, they have mentioned C12H24O12, which is not possible because, as I mentioned, when two molecules combine, a water molecule is released. And if a water molecule is released, that means two hydrogens are lost. Therefore, it cannot be H24, it should be H22. And no matter the disaccharide, the molecular formula is the same for both maltose and sucrose. Thus, the answer is A. Question 10. The diagram shows the structure of the polysaccharide chitin which is found in the cell wall of fungi. Which statements are correct for chitin and for cellulose? Statement 1. the monomers are joined by 1,4-glycosidic bonds. Statement 2, every second monosaccharide in the polysaccharide chain is rotated by 180 degrees. Statement 3, the polysaccharide contains the elements carbon, hydrogen, oxygen and nitrogen. Statement 3 is obviously incorrect because cellulose, they don't have the element nitrogen. Cellulose is only made up of chains of beta glucose, which consists of carbon, hydrogen and oxygen only. There is no nitrogen present in cellulose. And since the question asks which is correct for both chitin and for cellulose, statement 3 is incorrect. And since answers A, C and D have statement 3 in it, they all are incorrect, leaving us with the answer being only answer B, 1 and 2 only. Yes, answer B is correct because statement 1, the monomers are joined by 1,4-glycosidic bonds, is obviously shown in both cellulose and chitin. If you look at the diagram over here, I have labeled the carbon atoms from 1 to 4 of the middle carbon ring, and yes, bonds are formed between carbon 1 and carbon 4 of two neighboring carbon rings. Therefore, statement 1 is correct. Statement 2, every second monosaccharide is rotated by 180 degrees. Yes, that can be seen over here because the first and last carbon ring both have the sideways chain branching downwards, whereas the carbon ring in the middle has the side chain branching upwards. That means it must have been rotated by 180 degrees. And yes, obviously in cellulose, since it is formed from beta glucose units, they form linkages where every second monosaccharide is rotated by 180 degrees. Therefore, yes, statements 1 and 2 are correct, and hence the answer is B. Question 11. Which row correctly matches the functional and structural features of cellulose, collagen, glycogen or triglyceride? The molecules they have given are cellulose, triglyceride, collagen and glycogen. Key points you must remember are that cellulose and collagen are fibrous molecules, cellulose and collagen are unbranched, and triglycerides have no hydrogen bonds, whereas cellulose and collagen are held together by hydrogen bonds. And if you look at row A does seem to be correct. Cellulose is fibrous, which is correct. Molecules held together by hydrogen bonds is also correct. Branch chain should be false, which is also correct over here. Triglyceride is used as an energy source. Yes, we all know that because it can be stored in the form of fats, which is another energy source. Triglyceride is not fibrous. That is also correct. Molecules held together by hydrogen bonds, there is a false over here. That means that is also correct because triglycerides have no hydrogen bonds. And there is another false under branched chains because triglycerides are not branched. Hence, row A does seem to be the right answer. Let's look at row B. Why is row B incorrect? Well, as I mentioned, cellulose is held together by hydrogen bonds. However, under the column molecules held together by hydrogen bonds, they have put false for cellulose, which cannot be true because cellulose do have hydrogen bonds holding them together. Therefore, row B is incorrect. Row C. Why is row C incorrect? Well, under the column branch chains for collagen, they have put true. However, as I mentioned before, collagen and cellulose, they both are unbranched. Therefore, collagen is not having branched chains. Whereas over here, they have put true. Hence, row C is also incorrect. Why is row D incorrect? Because under the column molecules held together by hydrogen bonds for triglyceride, they have put true. However, triglycerides have no hydrogen bonds. Therefore, row D is also incorrect, leaving us with the only possible answer being row A. Question 12. A polypeptide has a number of amino acids N. How many peptide bonds in R groups, side chains, does this polypeptide have? Over here, for every pair of amino acids, one peptide bond is involved. Because if you look at your textbook diagrams, or even if you Google it, two amino acids, when they bond together, only one bond is involved, which is the bond in the middle. One bond is involved for every pair of amino acids. And since there are n number of amino acids, how many peptide bonds would that be? That would be n minus 1, not n peptide bonds, because every pair, one peptide bond is involved. So that would be n minus 1, leaving us with the answer being either A or B. And each amino acid has only one R group. So the polypeptide will also have n R groups because nothing happens to the R groups. It doesn't reduce by one, nothing happens to it. Therefore, the answer over here is answer B. N-1 peptide bonds and N R groups, because each amino acid has one R group, and for the number of amino acids N, it will have N R groups. Therefore, the answer over here is B. Question 13. What are the features of triglycerides? The columns given are polar, less dense than water, lower proportion of hydrogen than in carbohydrates. Triglycerides are non-polar, as they are hydrophobic or water-hating. Polar means they love water. However, triglycerides, they don't love water, they hate it. That's why they are known as hydrophobic in nature, or non-polar in other words. Hence, under the column polar, it should be no, leaving us with the answer being either C or D. And obviously, triglycerides are less dense than water, which is why you can see droplets of oil floating on water if you have carried out this experiment. Therefore, under the column less dense than water, it should be yes, leaving us with the only possible answer being C. Nonetheless, let's look at the column lower proportion of hydrogen than in carbohydrates. It should be false over here, because triglycerides has way too more hydrogens than carbohydrates due to the presence of many CH bonds in their fatty acids. It is because triglycerides have many fatty acid tails, and fatty acids do have lots of CH bonds or carbon hydrogen bonds. Therefore, the proportion of hydrogen is way too much compared to carbohydrates. Hence, it doesn't have a lower proportion than carbohydrates. In fact, it has a greater proportion. Therefore, the answer over here is C. Question 14. Which of these statements describe the action of an extracellular enzyme? 1. Synthesis of a polynucleotide in the nucleus during DNA replication. 2. Digestion of macromolecules in the lumen of the small intestine. 3. Synthesis of ATP molecules in the mitochondria. Statement 1. Synthesis of a polynucleotide in the nucleus during DNA replication doesn't involve an extracellular enzyme. DNA replication involves DNA polymerase, which is not extracellular. It is already present inside the nucleus. Therefore, statement 1 cannot describe the action of an extracellular enzyme. Statement 2. Digestion of macromolecules in the lumen of the small intestine. Yes, this can be a possible statement, because extracellular enzymes are those that are produced to carry out their functions elsewhere or outside from where it is made, which is evident in digestion of macromolecules in the small intestine, because some of the enzymes do come from other parts of the body and carry out their function in the small intestine. Therefore, yes, extracellular enzymes are obviously involved in digestion. Therefore, statement 2 does describe the action of an extracellular enzyme. Statement 3. Synthesis of ATP molecules in the mitochondria. Synthesis of ATP molecules is done by ATPase, which is an intracellular enzyme, not extracellular. Intracellular means an enzyme which performs its function wherever it is made. So if it is made in the mitochondria, it will perform its function in the mitochondria. So the only statement that describes the action of an extracellular enzyme over here is concerned with digestion, and that is statement 2 only. Hence the answer is D. Question 15. An experiment was carried out in which the enzyme lipase was used to hydrolyze a triglyceride. The pH was recorded at regular intervals during the reaction. The results are shown in the table. The table given over here has columns time, which is in minutes, and the pH. At 14 minutes, unreacted triglyceride was still present. What explains the results after 10 minutes? When a triglyceride is hydrolyzed, fatty acids and glycerol are produced, because triglycerides are formed by fatty acids and glycerol. So if a triglyceride is broken down, then obviously the products would be fatty acids and glycerol. Fatty acids being produced leads to a decrease in pH, because the word itself has acid in it. So acid means low pH, right? Thus over here, up to 10 minutes it decreases. Because fatty acids are being produced, it will become more and more acidic, hence the pH decreases. But after 10 minutes it stays constant. And why does it stay constant? That is what we have to find out over here. Since the pH is lowered from neutral, that means pH 7 is neutral. So if it is lowered from neutral to 4.6, that indicates the enzyme must have been denatured due to the extremes of pH, as enzymes work best at their optimum pH. So if the pH decreases rapidly from 7 to 4.6, that means it must have altered the enzyme. In other words, the enzyme must have been denatured. If the enzyme is denatured, that means the enzyme cannot carry out its function. And that could explain why the results after 10 minutes shows that the pH remains constant and unreacted triglyceride was still present at 14 minutes. As you can see over here, I have mentioned denatured means that the enzyme structure has been altered, and that means that the tertiary structure of enzyme had been lost. Change in enzyme structure is related to tertiary structure of an enzyme. So if there is a change in enzyme structure, that means the tertiary structure has been affected or altered. And if you look at the answers, A, the end products acted as competitive inhibitors. No, that cannot be true, and we cannot be certain of this answer. Answer B, the end products acted as non-competitive inhibitors. Again, we do not know if the end products acted as competitive or non-competitive. So answers A and B are not answers we must go for because we are not really sure about that. Answer C, the enzyme reaction had reached Vmax. Vmax means the maximum rate of reaction, and over here it has nothing to do with maximum rate of reaction because after 10 minutes the reaction seems to have stopped. So since the question asks what happens after 10 minutes, it cannot have reached maximum rate of reaction. In fact, the reaction has stopped. That is why the pH remains constant and there is unreacted triglyceride left at 14 minutes. Therefore, the only answer that must be correct over here is answer D. The tertiary structure of the enzyme had been lost. Yes, because as I mentioned, the pH is lowered from 7 to 4.6. That has caused the enzyme to be altered or denatured because enzymes cannot work at such extremes of pH, as enzymes work best at their optimum pH. If the enzyme has been denatured, that means the enzyme structure has been altered, and that means the tertiary structure had been lost. Hence the answer is D. Question 16. Which statement about the effect of substrate concentration on the activity of an enzyme is correct? A. Above a certain concentration of substrate, an enzyme reaches its maximum rate of reaction. Well, if you're not sure about this answer, do keep this aside and consider the other options. However, if you have revised your notes and if you have studied the topic regarding enzymes, you should know that maximum rate of reaction or Vmax is indeed reached above a certain substrate concentration, and that you can refer by looking at graphs in your textbook or even just researching online. So if you are not sure about this answer, leave it aside and consider the other answers. Answer B. At high concentration of competitive inhibitor, increase in the substrate concentration has no effect. Competitive inhibitors are affected by substrate concentration, as increasing substrate concentration does have an effect on competitive inhibitors. Hence B is incorrect because B mentions that there is no effect. However, it is the opposite. Increase in the substrate concentration does have an effect on the competitive inhibitor's function. Therefore, statement B is incorrect. Statement C. At high substrate concentration, a non-competitive inhibitor no longer affects the enzyme activity. Non-competitive inhibitor is unaffected by changes in substrate concentration. Hence, will affect the enzyme no matter the substrate concentration. Therefore, C is incorrect. What I mean by this is, non-competitive inhibitor, it is not affected by changes in substrate concentration. So, no matter if it is high substrate concentration or low substrate concentration, a non-competitive inhibitor will always continue to carry out its function. Therefore, statement C is also incorrect. Statement D. The higher the concentration of substrate, the faster an enzyme can catalyze a reaction. Speed of an enzyme catalyzed reaction doesn't depend on substrate concentration at all, but it depends on the presence of a catalyst, because a catalyst speeds up the rate of an enzyme reaction. Hence, statement D is incorrect. And since we have ruled out statements B, C and D, the only possible answer over here is statement A. So if you didn't know the answer at first by looking at it, we have reached the answer by just cancelling out the other options. And now you know that maximum rate of reaction is reached above a certain substrate concentration. Keep this in mind. Hence the answer is A. Question 17. Which row shows a correct function of the components of the cell surface membrane? The components given are phospholipids, cholesterol, proteins and glycoproteins. Let's look at row A. Phospholipids prevent the entry of non-polar molecules. That is incorrect because phospholipids, they always prevent the entry of polar molecules. Do keep in mind this point. Therefore, row A is incorrect. Row B. Prevent the entry of polar molecules. Yes, that is correct. Cholesterol stabilizes the membrane by binding to water molecules. Cholesterol never binds to water molecules because it is water hating in nature. Do keep this point in mind. Therefore, row B is incorrect. Let's look at row C. Row C is also incorrect by just looking at one certain column. And what is that column? Proteins. It has mentioned proteins allow exchange of gases between the cell and its environment. Exchange of gases occurs by diffusion, and diffusion doesn't require any sort of proteins. It doesn't require any transport proteins or carrier proteins, nothing. Therefore, proteins is not involved in exchange of gases. Therefore, row C is incorrect. Hence, that leaves us with row D being the only possible answer that shows a correct function to its cell components. Phospholipids regulate the fluidity of the cell membrane. Yes, it is correct, because phospholipids, they are concerned with stability, fluidity and flexibility of the cell surface membrane. So yes, it does regulate the fluidity of the cell surface membrane. Cholesterol stabilizes the membrane over a range of temperatures. Yes, that is correct. That is also another function of cholesterol. It stabilizes the membrane over a range of temperatures. Do keep these points in mind. Proteins form channels for polar molecules. Yes, that is correct, because polar molecules, as I mentioned before, they cannot enter the phospholipids due to the phospholipids having a hydrophobic region. Hence, proteins must be present to allow the polar molecules to travel through the cell surface membrane. Otherwise, how will the polar molecules enter the cell? So, proteins do form channels for polar molecules. And finally, glycoproteins allow cell-to-cell recognition. Yes, that is correct, because a function of glycoproteins is cell-to-cell recognition or even in cell signaling functions. Hence, the answer over here is row D. Question 18. The graph shows how the rate of entry of substance X into a cell changes as the concentration of substance X outside the cell increases. The diagram shows part of a cell surface membrane. Which pathways could substance X use to enter the cell? So from the graph itself, it can be seen that diffusion occurs, because as the concentration of substance X outside the cell increases, the rate of entry also increases, then reaches a plateau, or the gradient levels off, it becomes horizontal. That means there is no concentration gradient when there is a plateau on the graph. No concentration on the gradient due to the plateau at the end of the graph. Hence, this is diffusion, because diffusion only occurs if there is a concentration gradient difference. If the concentration outside and inside becomes equal, then the rate of entry reaches a plateau. So this has to be diffusion. Now we know that diffusion is a possible pathway. And what does diffusion basically do? Diffusion is a movement from high concentration to low concentration, that is down a concentration gradient, which doesn't require any sort of energy. It is a passive pathway. So, pathway 1 is definitely diffusion. Pathway 2 is facilitated diffusion because it uses a channel protein. And both facilitated diffusion and diffusion is where the movement occurs from a high concentration to low concentration, that is down a concentration gradient. So, the pathways where substance X could use to enter the cell is 1 and 2 only. Why not 3? Because 3 represents active transport. As you can see, it is through the use of a transport protein, and active transport is not the graph plotted over here, whereas it is diffusion. So facilitated diffusion and diffusion can both occur because they both are concerned with movements from high concentration to low concentration down a concentration gradient, and it doesn't require any ATP or energy. Therefore, the pathways are 1 and 2 only, which is answer A. Question 19. The photomicrograph shows cells in various stages of the cell cycle. In which stage does semi-conservative replication of DNA take place? Stages A, B and D are part of mitosis, whereas C is not part of mitosis, but C represents DNA replication. So the stage semi-conservative replication of DNA occurs in the photomicrograph labeled as C, because A, B and D shows mitosis. A is for metaphase, B is for anaphase and D is for prophase. C can only be seen during DNA replication. C is not part of mitosis. Therefore, the stage that shows semi-conservative replication of DNA is stage C. Question 20. The diagram shows the structure of one chromosome. Which row is correct? I have labeled over here what the parts of a chromosome are, So 1 represents a centromere. It is what holds the two chromatids together, and 2 represents telomere. And remember, one chromosome is made up of 2 chromatids, and telomeres lies towards the end of a chromatid, as shown by number 2. So which row is correct? 1 is centromere, leaving us with the answer being either A or B, and 2 is telomere. So the answer must be answer B. And yes, the number of DNA strands would be 4. Why is that? Because one chromatid is made up of 2 DNA strands. Therefore, 2 chromatids would be made up of 4 DNA strands, because one chromosome, as shown over here, is made up of 2 chromatids. So 2 times 2 is 4, hence 4 DNA strands. Therefore, the row which is correct over here is row B. Question 21. Cancer cells may divide by far more divisions than other cells found in humans. Which statement about cancer cells is correct? A key thing you must know is that cancer cells are able to divide repeatedly and undergo multiple cell divisions due to the fact that they are able to synthesize telomerase, which is an enzyme that replaces telomeres, because without telomeres the cell cycle stops. Telomeres are the only ones which aid the cancer cells to keep on dividing and dividing. So the answer over here must be answer A. They are able to synthesize the enzyme telomerase. It is because telomeres, they start with a fixed number of DNA sequences in the beginning, and then with each cell division some of the telomeres are lost. If the telomeres are not replaced, then the cancer cells are not able to divide repeatedly. However, the purpose of cancer cells is they keep on dividing far more rapidly than other cells in humans by mitosis, and that is done because cancer cells have the enzyme telomerase, which replaces the lost DNA sequences. Hence, the statement over here that is correct is statement A. They are able to synthesize the enzyme telomerase. The other statements are incorrect because they do not talk about the role of cancer cells dividing, because cancer cells, they undergo multiple cell divisions, and that can only be explained by statement A. Statement B, C and D do not explain why cancer cells may divide by far more divisions than other cells found in humans. So, the key part of this question is the first statement. Cancer cells may divide by far more divisions than other cells found in humans. From that, you must realize that it is concerned with telomeres and the enzyme telomerase. Therefore, always look out for the answer which has a point relating to this. So, the statement about cancer cells which is correct over here is only statement A. And regarding cancer cells, all you have to know is this statement. They are able to divide far more rapidly than other cells in humans by mitosis. They undergo multiple cell divisions due to the fact that they are able to synthesize telomerase, which is an enzyme that replaces telomeres. Therefore, what is correct over here is statement A. Question 22. What is the maximum number of hydrogen bonds in the length of DNA containing 700 nucleotides? Hydrogen bonds are always formed between two nucleotides or a pair of nucleotides. Hence, over here, if there are 700 nucleotides, that means there are 350 pairs. How do you find out how many pairs are there? Well, 700 divided by 2 will give you the number of pairs. So that is 350 pairs. And since the question refers to maximum, which is highlighted over here, the word maximum is in bold. Hence, you should always go for 3 bonds being present between two nucleotides. That is, for each pair there will be 3 bonds, because it refers to maximum number of hydrogen bonds. Do remember that there are 4 types of nucleotides in DNA: A, T, C and G. So A binds with T using 2 bonds and C binds with G using 3 bonds. Since the question refers to maximum, we have to assume that all of the nucleotides in this DNA containing 700 nucleotides will have either the base C or the base G. So if we assume it to have all being either C or G, then all of them will have 3 bonds between 2 nucleotides. Therefore, first we get the number of pairs present, that is 350 pairs, then we multiply it by the number of hydrogen bonds. If it is maximum, you multiply it by 3. If it is minimum, you multiply it by 2. Since the question is maximum, multiply 350 by 3, you will get the answer as 1050. So the answer over here is C. Question 23. Which statements concerning DNA and RNA are correct? 1. Adenine and guanine are bases that have a double ring structure. Cytosine, thymine and uracil are bases with a single ring structure. Yes, that is correct. Because adenine and guanine, well, they are purines. Remember the word Gapur, where guanine and adenine are purines. And purines have double ring structure, whereas pyrimidines have single ring. This is the only point which you have to remember in this statement. Purines have double ring and pyrimidines have single ring. How do you know the purines present? Just remember the word Gapur, G-A-P-U-R, where G is guanine and A is adenine. So guanine and adenine are purines, which is represented by PUR in the word Gapur. Therefore, yes, statement 1 is correct. Statement 2, an adenine nucleotide from DNA is the same as an adenine nucleotide from RNA. DNA adenine pairs with thymine and RNA adenine pairs with uracil. Also, remember, any DNA nucleotide, which over here is adenine nucleotide, is always different from the equivalent RNA nucleotide. So, an adenine nucleotide in DNA is not the same as the adenine nucleotide from RNA. Why is that? Because DNA has the sugar deoxyribose and RNA has the sugar ribose sugar. Therefore, they won't be the same. They will have some sort of difference. Therefore, statement 2 is incorrect. And if you go through the answers A, B, C and D, answers A, B and D have statement 2 in it. Therefore, leaving us with the only answer being answer C. Because statement 2 is incorrect, therefore we cannot choose answers A, B and D. Answer C, however, 1, 3 and 4, yes, we can choose it. Nonetheless, let's look at statements 3 and 4. 3. The base pairing that occurs in the double DNA helix and when RNA is synthesized during transcription is always according to the rule that a purine pairs with a pyrimidine. Yes, this is correct, because during base pairing, a purine always pairs with a pyrimidine. Do keep this point in mind. And statement 4, the two polynucleotides on a DNA molecule run in opposite directions so that the double helix formed has two strands that are parallel to each other. Yes, opposite directions but parallel to each other is referred to as the anti-parallel nature, or they are anti-parallel strands, which is present in a DNA molecule. Hence the answer is C, 1, 3 and 4. Question 24. Two sets of bacteria were grown using different types of nitrogen containing growth media. One set was grown in a medium containing the heavy isotope of nitrogen 15N until all the DNA was labeled. The other set was grown in a medium containing the light isotope of nitrogen 14N until all the DNA was labeled. The DNA from each set of bacteria was extracted and centrifuged. The diagram shows the position in the centrifuge tubes of this DNA. Bacteria with 15N labeled DNA were transferred to a medium containing 14N and allowed to reproduce once. The DNA of the new generation of bacteria was extracted and centrifuged. Which tube shows the position of DNA from this new generation of bacteria? So this is a long question. However, key points you must know is that this test is there in your textbooks as well. And basically, first they grow bacteria in a medium containing the heavy isotope of nitrogen, that is 15N. So first, all the DNA would consist of 15N. Then, the bacteria labeled as 15N were transferred to a medium containing 14N, and 14N means the light nitrogen. So, all the bacteria which was grown in 15N was transferred to 14N and allowed to reproduce once. After transferring 15N to 14N and allowed to reproduce once, you would get the first generation of bacteria, and we are supposed to find out where the position of this DNA would be in a tube. So since this question is regarding the first generation would always have 100% hybrid DNA. That is, each DNA molecule will have one 15 nitrogen strand and one 14 nitrogen strand. So always remember, first generation of bacteria obtained where both are allowed to reproduce once would be 100% hybrid. 100% hybrid means each DNA molecule will have one 15N strand and one 14N strand. Hence the position would be towards the middle of the test tube, which is only shown by test tube D, because it is 100% hybrid, so there can only be one position in a test tube. Answers A and C are incorrect because there are two positions, which is not correct if it is 100% hybrid. Answer B is incorrect because answer B resembles that of DNA with 14N. However, since both are mixed and both will have contributed towards forming a new DNA molecule with both 14N and 15N, it has to be towards the middle because 15N lies at the bottom and 14N lies at the top. So once they mix and form 100% hybrid, that will be in the middle of the test tube. Hence the answer over here is answer D. and this position is only possible if it is allowed to reproduce once. Do keep this point in mind. If it is allowed to reproduce twice, it would be a different position obtained. Question 25. The diagram shows a nucleotide sequence of a small section of a gene which is transcribed. The table shows the amino acids coded for by 10 mRNA codons. What is the order of the 4 amino acids in the polypeptide translated from this small section of a gene? Shown is a nucleotide sequence of a gene. That means before the process of transcription has occurred. Since the table shows that of mRNA codons, first you have to convert the entire sequence given into a mRNA strand. How do you do that? Well, you should know that when transcription occurs, complementary base pairing occurs. So if the DNA sequence starts with G, then the mRNA strand would start with C. And what is the complementary base pairing rule? C binds with G and A binds with T or U. So in this given gene sequence, you can only see C and G. So we have to consider the base pairing rule, that is C binding with G. As you can see over here, I have mentioned what the mRNA strand is. It is CGC, GCG, CCG, CGC. How did I obtain that? Just match it using the base pairing rule. C binds with G, and it should be opposite in nature to that of the DNA sequence. Now that you have got the mRNA strand, you have to match it with the table given over here, because the table has the mRNA codons which we just found out, and we have to match the amino acids because the question asks what is the order of the 4 amino acids in the polypeptide translated from this small section of a gene. Since the mRNA strand starts with CGC, the corresponding amino acid would be ARG, leaving us with the answer being either C or D. Let's look at the next codon, GCG. Where does GCG lie in this table? Well, as you can see, GCG matches with ALA. So the answer over here is definitely C, not D, because D has ARG. However, it should be ARG followed by ALA, not another ARG. Therefore, the answer over here is answer C. Question 26. What contributes to the movement of water through the xylem vessel elements? Movement of water through xylem is mainly due to cohesion and adhesion forces. So if you see this kind of question, you have to look out for the words cohesion and adhesion. So let's look at answer A. Cohesion of water molecules through hydrogen bonding. Yes, this must be the answer, because answers B, C and D do not talk about cohesion and adhesion. Why are answers B, C, D incorrect? Because it doesn't contribute to the movement of water. Movement of water is mainly concerned with cohesion and adhesion only. Do keep these points in mind. So the answer over here is answer A, cohesion of water molecules through hydrogen bonding. And yes, because cohesion does occur by hydrogen bonding of neighboring water molecules. Question 27. The diagram shows a xerophytic leaf in different conditions P and Q. Which statements about the cells in layer X of the leaf in each of the conditions P and Q are correct? Statement 1. Less negative water potential in P than Q. The leaf in condition P obviously has a higher water potential than the leaf in condition Q, because Q shows a leaf which is curled up. Curled up means lower water potential present, and that means less turgid it is. A key point you must also remember is that less negative means higher water potential and more negative means lower water potential. An easier way to picture this would be to remember the number scale, where 0 is the highest water potential possible, because pure water has a water potential of 0. So from the scale, the more negative the number becomes, that means going further down the scale away from number 0, the number becomes more negative. And since 0 has the highest water potential, the more negative the number becomes, the lower the water potential. However, the less negative the number becomes, that means reaching towards 0, then the water potential increases, or the higher the water potential becomes, because 0 is the highest water potential possible in any cell. So obviously statement 1 is correct. Less negative water potential in P than Q, because P shows a leaf with a higher water potential than Q. So 1 is correct. 2. Cells may be turgid in P and plasmolized in Q. This is another possibility, because higher water potential means more turgid the leaf is. So cells in P can be turgid, whereas cells in Q can be plasmolized. Plasmolysis occurs in regions where there is very low water potential. So Indeed, Q shows a leaf placed in a condition with very low water potential. Therefore, some cells may be plasmolysed. So statement 2 is also correct. Statement 3, cells less turgid in P than Q. That is obviously incorrect because P shows higher water potential. Higher water potential in P doesn't mean less turgid, but it means more turgid. So statement 3 is incorrect. Statement 4, no net diffusion of water into X in either P or Q. Now this statement is a tricky statement. Why is that? The statement mentions net diffusion of water. However, we know that water doesn't diffuse in a leaf or inside cells, but water follows osmosis. So it should be over here no net osmosis, not diffusion. Although you might think that statement 4 is incorrect, it is correct. Why is that? Because they have mentioned no net diffusion of water. And yes, inside cells in a leaf, water doesn't diffuse, but water follows down a water potential gradient by osmosis, not diffusion. So yes, 4 is correct. Hence the answer over here is answer B. 1, 2 and 4 are the correct statements. Question 28. What are features that adapt root hair cells for efficient uptake of mineral ions? 1. A large number of mitochondria. Yes, mineral ions enter through active transport, which requires a lot of energy and transport proteins in membrane. Energy is provided by mitochondria, and hence root hair cells do need a large number of mitochondria for its efficient uptake. Feature 2. A large number of protein carriers in the cell surface membrane. Yes, because active transport occurs, active transport requires carrier proteins, therefore large number of protein carriers is required. Feature 3. A large surface area to volume ratio. Root hairs indeed do provide large surface area to volume ratio. Hence the answer over here is answer A. 1, 2 and 3. Question 29. The transverse sections of two plant organs are shown. Which pair of tissues contains proton pumps? I have labeled over here which one represents the xylem and which one represents the phloem. So Q and T represents xylem, R and S represents phloem. And phloem is required in translocation of sucrose, and translocation of sucrose obviously does require proton pumps. So proton pumps are only required in phloem tissue. And which one shows phloem tissue over here? R and S. Therefore the answer over here is C, R and S. The other answers are incorrect because they mention a xylem tissue, whereas inside a xylem only water movement occurs. There is no translocation of sucrose, hence no proton pumps are required in xylem. It is only required in phloem tissue. Question 30. Measurements of the stem of a young tree show that the diameter varied slightly. It decreased during the day and increased during the night. Which statement explains these observations? Always in these kind of questions, if they talk about the stem of a tree, that means it is referring to the diameter of a tree. And if the question gives you the slightest hint that it mentions about the diameter of a tree, look out for the word tension in the answers. So, which statement explains these observations? You have to look out for the answer which has the word tension in it. Let's look at answer A. During the day, cohesion between water molecules and lignin in the walls of the xylem vessels pulls the walls of the vessel inwards. That doesn't talk about tension. So statement A is incorrect. Statement B. During the day, the tension developed in a moving column of water molecules reduces the pressure inside the xylem vessels so that their walls move inwards. Yes, this is correct. And this is the only answer that has the word tension in it. So in these kind of questions talking about the stem of a young tree where the diameter is varied, you should look out for answers related to tension. So the answer over here is answer B. If you go to the answer C and D, they do not have anything relating to tension, hence they are incorrect explanations. Because once tension develops in a moving column of water, pressure is reduced inside the xylem vessels as statement B explains. So if the pressure inside xylem vessels is reduced, the walls move inwards. That is why it decreases during the day. At night, the tension is not there, so the pressure increases. If the pressure inside the xylem vessel increases, then the walls move outwards. That is why the diameter increases during the night. So always in these kind of questions, look out for the word tension as I mentioned. Question 31. Which statement about the human circulatory system is correct? A. Blood passes twice through the heart in one complete circulation. That is a correct statement. So let's keep this statement aside and consider the other statements. B. Blood, tissue fluid and lymph are all parts of the circulatory system. Lymph doesn't play a part in circulatory system. It plays a part in the lymphatic system. Hence B is incorrect. C. Capillaries have the lowest blood pressure. That is incorrect. Veins have the lowest blood pressure, not the capillaries. Therefore statement C is also incorrect. D. Veins in the circulatory system all carry deoxygenated blood. We might think this is true, but not all veins carry deoxygenated blood. An example would be the pulmonary vein. Pulmonary veins carry oxygenated blood, not deoxygenated blood. Therefore statement D is also incorrect, leaving us with the only possible answer being statement A. Blood does pass twice through the heart in one complete circulation. That is why humans have double circulation or a double circulatory system. Hence answer is A. Question 32. The graph shows changes in the volume of the ventricles during the cardiac cycle. Which valves open and close at P and R? The graph shows the volume in ventricles only, and that is mentioned by the question given over here. Graph shows changes in the volume of ventricles. So this is regarding the ventricles only. At P, it is almost a start of the ventricular systole, where the ventricular contraction occurs. Systole means contraction, diastole means relaxation. Do keep this point in mind. So ventricular systole occurs at P, where ventricular contraction occurs. Hence at P, semilunar valves open, because when there is ventricular contraction, semilunar valves would open and the atrioventricular valves closes. If you are not thorough with this section, I would advise you to go revise through these points in your textbook. Again, ventricular contraction, semilunar valves open and atrioventricular valves closes. So at P, the atrioventricular valve should close, leaving us with the answer being either A or B. Let's look at R. At R, almost it is the start of the ventricular diastole, meaning relaxation of the ventricles. As I mentioned, diastole means relaxation. So if the ventricles are being relaxed, then the semilunar valves closes and the atrioventricular valves open to allow the blood to enter the ventricles. Therefore at R, does the semilunar valve close or open? The semilunar valve closes. Hence the answer over here is answer A. Atrioventricular valve at P closes, semilunar valve at R also closes. Question 33. Hemoglobin can bind to carbon dioxide, carbon monoxide and oxygen. Which gases share a binding site? Although all can bind with hemoglobin, only carbon monoxide and oxygen share a binding site with hemoglobin. Do remember this point. Again, only carbon monoxide and oxygen share a binding site. So the answer over here is answer D, 2 and 3 only. Carbon dioxide does not have a binding site in hemoglobin. It is only carbon monoxide and oxygen. So the answer over here is again answer D. Question 34. A good gas exchange system maintains a steep diffusion gradient, has a large total surface area and a short diffusion distance. Which feature of the human gas exchange system helps to maintain a steep diffusion gradient? Steep diffusion gradient is related to the difference in the concentration of gases. So if you go to the answers A, B, C and D, the answer that talks about a difference in the concentration of gases would be answer C. The air brought into the alveoli by ventilation is high in oxygen. In order for alveoli to obtain oxygen into our blood, there should be a concentration difference. That means the air brought into our alveoli by ventilation should be high in oxygen, because diffusion occurs from regions of high concentration to low concentration. So high concentration would be the air brought into the alveoli, and low concentration is the blood inside our capillaries. And what helps to maintain this steep diffusion gradient is ventilation. So in this kind of question, look out for the word ventilation, and if you see an answer which is regarding ventilation, then that is the correct feature. The feature of the human gas exchange system that helps to maintain a steep diffusion gradient is concerned with the difference in concentration of gases, and that is only brought about by ventilation. Hence the answer is C. Answers A, B and D do not talk about ventilation or concentration gas difference. Question 35. Which statement about bronchioles is correct? A. They have a cartilage and ciliated cells. B. They have cartilage and elastic tissue. C. They have cartilage and muscle tissue. D. They have elastic tissue and ciliated cells. Answers A, B and C all have the word cartilage. But do bronchioles have cartilage? The answer is no. Bronchioles do not have any cartilage at all. Only trachea and bronchus have cartilage. Remember this. So, bronchioles do not have cartilage. Hence, the only possible answer over here is answer D. They have elastic tissue and ciliated cells. Question 36. In which regions of the human gas exchange system is the function of some types of cell directly affected by tar and cigarette smoke? Tar directly affects the ciliated epithelium. Always, if the question is concerned with which cell is affected by tar, tar affects the ciliated epithelium, and ciliated epithelium is only present in trachea, bronchus and bronchioles. Why not alveoli? Because alveoli do not have ciliated epithelium. They have a different type of epithelium known as the squamous epithelium. So, the answer cannot have alveoli in it, but the answer should have trachea, bronchus and bronchioles. So, the function of the cell types directly affected by tar would be trachea, bronchus and bronchioles. So, the answer over here is answer D. Trachea, bronchus and bronchioles only. Question 37. What is a causative agent and method of transmission of smallpox? Smallpox is caused by a virus known as the variola virus. The method of transmission is through direct contact. It is not waterborne. So the answer over here is answer C. The causative agent is variola, not morbillivirus. Morbillivirus is the causative agent of measles. Smallpox is caused by variola virus. So the answer is C, because the causative agent is variola and the method of transmission is direct contact, not waterborne. Question 38. A scientist investigated the effect of an antibiotic on the treatment of cholera. 320 people suffering with cholera were split into two groups. One group was treated with an antibiotic while the other group was not given antibiotics. Both groups were given fluids containing sugars and mineral salts, also known as oral rehydration therapy. The scientists recorded the number of days that each person suffered from diarrhea. The table shows the results. What is the percentage decrease in the mean time that a person suffered from diarrhea when they were treated with the antibiotic? Percentage decrease is the difference upon the starting initial value from which it decreased, and that answer into 100. So, percentage decrease would always happen from a higher number to a lower number. So, the initial number would be 5.3. and the decrease would be 5.3 minus 3.2. So 5.3 minus 3.2, that is the difference upon the starting initial value from which it decreased. So from which number did it decrease? It decreased from 5.3. So the difference, which is 5.3 minus 3.2 over the initial value, which is 5.3, that answer into 100 would give you the percentage decrease. Therefore, if you carry out the math, you get the percentage decrease as 39.6%. That means the answer is A. This question has nothing to do with biology but just applying math. Question 39. Which sequence of events occurs during an immune response? Immune response always starts with the recognition of non-self antigens or foreign antigens. So, sequence of events should start from 3. Leaving us with the answer being either C or D. 3 talks about the recognition of non-self antigens, which occurs first. Recognition of non-self antigens or foreign antigens leads to mitosis of B lymphocytes. So the second step would be mitosis of B lymphocytes. That means event 2 occurs next, leaving us with the only possible answer being D, because C talks about event 1 following event 3. However, event 1 does not follow event 3. It should be event 2 which follows event 3. So the answer over here is D. Nonetheless, in an immune response, first there would be the recognition of foreign antigens or non-self antigens. This leads to mitosis of B lymphocytes, which forms plasma and memory cells. B lymphocytes, when they undergo mitosis, they can form plasma and memory cells. Plasma cells are the only cells that secrete antibodies. So the secretion of antibodies occurs last. So the sequence of events are 3 followed by 2, 2 followed by 1, and 1 followed by 4. Question 40. An influenza vaccine can be made by growing the viruses in chicken eggs. The viruses are extracted in liquid from the eggs and inactivated. The purified egg extract containing the viruses is then used as a vaccine. What is the side effect of using this vaccine in some people? The liquid in chicken eggs could contain non-self antigens that causes an immune response, as the body doesn't recognize non-self antigens. As we discussed in the previous question, non-self antigens leads to an immune response. So, the side effect of using this vaccine in some people would be an immune response to egg antigens could occur. Because egg antigens are non-self antigens or they are foreign antigens. If they are injected into a person, the person won't recognize these egg antigens because they are from a chicken. So these would obviously be non-self antigens or foreign antigens, hence an immune response would occur, and that is a side effect. So the only answer over here is answer B. An immune response to egg antigens could occur. And that is the end of this paper. Hope you found this video useful. If you did, don't forget to leave a like and also do comment from which country you're watching this video right now. Thank you for watching and stay tuned for more.